Somewhere before now, you have seen that statement, the central dogma of biology. What is it? We can just start with the simple one. Low-hanging fruit. What's a dogma? Not necessarily a science term. Law, core foundational belief, right? Like something you hold to be true in your worldview or vision. So that core belief at the very center of biology says that all of our life information is based in what molecule? And that DNA really just serves as a code for what other nucleic acid? And really, all that RNA does is codes for protein. Everything that we believe in biology is based on this principle, this idea. DNA is going to code for RNA, and RNA codes for protein. It's pretty simple. How many of you have a birth certificate? Excellent. You were all, in fact, born. How many of you have a Social Security card? How many of you are like, yeah, I have one somewhere? Excellent. It probably applies to that birth certificate question too, right? You're like, yeah, I have one somewhere. Do you carry it around with you? Either of those pieces of documentation? No? Why not? What if somebody asks you who you are? So you'd use a driver's license instead?
Tell them who you are.
Tell them who you are. What if they ask for proof?
School ID.
School ID? Like one of those, you guys have those like weird ID cards that go in the wrong direction for those of you who have a state identification card. And when you get your driver's license, you'll get the weird driver's license that goes the wrong direction too. So we all know that you're under 21? And then you get like the normal direction driver's license. Isn't that right? Sounds vaguely familiar. I don't know. They started that weird direction thing long after I had a driver's license. So I don't know what's up with that. But why don't you carry your Social Security card or birth certificate on your person?
Is that necessary?
Is your identification necessary? Is it important for people to know who you are? Did the school have to have some kind of documentation of who you are before we admitted you?
Yes.
Absolutely. Do public schools require some kind of documentation of who you are before they admit you? What do parents send to school then? If they don't send you with your birth certificate or with your driver's license or with your Social Security card, what do they send?
Copy?
Why would they send a copy?
Because they have the actual, if they give you the actual one, they have one for others.
So there are multiple reasons you're going to need both of those documents in your life. What happens if they gave you the original and sent you to school with it, and you had a moment and it went missing? Your parents are going to flip out. Why would they flip out? So it's easily stolen. And those are particularly valuable documents. Do you suppose people steal those documents because they're valuable documents? How many of you know where those things are located in your house? Is it in a small metal box?
Yeah.
Is it in a safe? Or a small metal safe, as it may be, or some of you have it in a small metal box in a larger metal safe. That's like protection upon protection. Some of you have a large safe with it in it. So we keep it in a protected, those small metal boxes are called fire boxes. Because if everything else around it should happen to burn, the contents of the firebox does not. So anything that's in there, it won't burn. We keep it someplace safe because it is exceptionally valuable, and when you need it, you are only given a copy. And your parents can make as many copies as you need, because when they submit a copy of your birth certificate to the school does not give it back. The school keeps it. And any other time you're going to need a copy of your birth certificate, you are not going to get it back. The people requesting it are going to keep it. So you have to be able to make multiple copies of the original. And it makes sense why. The original is valuable. You're not going to give that to just anybody, and you want to make sure nothing happens to it. We keep the original someplace very safe, someplace that it never leaves. And it will always be used there. And copies can be made of it. And that copy is just a copy. It's temporary, it's cheap, it's disposable, and it's damageable. It's cheap and replaceable. And the process by which we make that copy is simply called transcription. You guys know that I like to look at parts of words and what they mean. Trans means across, right? Scribe means to write. So we're simply writing a copy that we can send across our cell membrane or across that nuclear membrane. It's a cheap temporary copy, and it's transcribed. And that's always in the nucleus. RNA, it gets to leave. It gets to go to all of those places that it's needed, which typically are out in the cytoplasm. Makes sense. We get why we would do that. RNA is not the final product, though. The thing that makes your body do all the things that your body does is protein. Hopefully you remember protein, the monomer is a what? Amino acid. And how many of them are there?
20.
So somehow we have to change languages from the language of nucleic acid to the language of amino acid. You guys do that all the time in your foreign language classes, right? You're given a text in, say, Latin, and you're asked to make it English. Do you agree? What do you call that? It's a translation. We'll do the same thing here. We translate. Except here, instead of going from Latin to English, or English to Latin, or Spanish to English, English to Spanish, here we go from nucleic acid to amino acid. We just change the alphabet we use. Not a big deal. These two processes added together are called protein synthesis. Those two things added together are called protein synthesis. The two steps themselves, super simple. Not especially complicated. Easiest place to start is by making a nice comparison of things you already know. I'm going to have you and your partner fill these things out for DNA. Let me hear him.
For this shape, I got a double helix. For the number, I have, for the number, I have 2. For the basis, I have A, T, C, and G. For the sugar, deoxyribose.
Absolutely correct. Our friend RNA then is not a double helix. It's a single strand. A single linear strand. So if it's a single linear strand, how many strands does it have?
One.
The nitrogen bases will be adenine, cytosine, and guanine with one change, uracil instead of thymine. The sugar is ribose rather than deoxyribose. Now, all this time we've been saying deoxyribose, we kind of have just been like mimicking some words, making some sounds, and sounding them out. But you know, scientists and our penchant for liking to take words apart and making sure that each part means something, D means what? Di means two, but D, if I, let's see. If something devolves into nonsense, what does it become? It goes down or backwards. To denude something means to remove all of the covering, right? To deflate means to take out. So D must mean without, and oxy must mean oxygen. The single difference between deoxyribose and ribose sugar, they're both 5-carbon sugars. They both have a 1, 2, 3, 4, 5 carbon. They can be labeled exactly the same way. The single tiny difference is that ribose has an extra oxygen attached to one of the carbons, and deoxyribose does not. One tiny atomic difference. Emily?
What's the U one for the?
Uracil. URACIL. Replaces thymine. And of course, RNA's location, we said before, it can kind of go wherever it wants, but mainly it's in the cytoplasm. What's up?
So, ribose has an extra oxygen and another carbon?
That seems like such an itty-bitty difference, but all the difference in the world. Then again, the loss of one oxygen is also what makes the difference between water and hydrogen peroxide. So I guess one atom, kind of, sort of a big deal. Okay, it's a pretty big deal. There's only one kind of DNA, it's the kind you're familiar with, but RNA comes in 3 types. Messenger RNA, transfer RNA, and ribosomal RNA. Scientists are lazy, right? So instead of messenger RNA, we simply write lowercase m, all capital RNA. Instead of T, or transfer RNA, it's a lowercase t, and then everything else capital RNA. And instead of ribosomal RNA, lowercase r, everything else capital RNA. Every single one of those forms of RNA plays a super important role in the processes of transcription and translation, but you'll learn them as we go rather than before we start. They all have a special job. The first process we
Okay, I'm good.
on, do I need to go back? Was that too fast? Are all our hands done writing? No? You need to go back then?
Yeah.
When we talk about transcription and translation, I often refer to cooking. Why? Well, one, cooking and baking is a hobby of mine, and two, to me it just kind of makes sense. How many of you have, this is a dying thing, I'm not sure how much longer these things are going to exist. How many of you have cookbooks in your house? When you need a recipe from the cookbook, in fact, if you take any of the classes with Mrs. Sovine, I know this is in fact true because I am often at the photocopier for students. You don't use the whole cookbook at once, right? How many recipes do you use at a time? One. And sometimes, I don't know, you know some cookbooks are really expensive, right? For example, Mastering the Art of French Cooking, massively expensive cookbook. And it's massively expensive cookbook that I own that I refuse to take in the kitchen. Then how does one go about using it? Yeah, you have to copy it. If I were to not copy the whole recipe, say it's one of those dumb recipes, actually they don't do this in Mastering the Art of French Cooking, they intentionally don't, but say it's a recipe that starts on the bottom of one page and continues on to the next page. What if I didn't copy the beginning? Yeah, I'm gonna just like jump in halfway through the procedure of making it, I'm not gonna have an ingredients list, it's gonna be a mess. Same thing is true for any region of DNA that codes for protein. Hey, is there a name for a region of DNA that codes for protein? It's a gene. It's a region of DNA that codes for a protein. So in order to make sure we get the whole recipe for that protein, to get the whole gene, we have to start at the beginning. And we do that pretty simply like this. Uh-oh. No, none of those are the one I want. Of DNA, agreed? It's a double helix, stretch of DNA. The gene is indicated right there. We actually start copying ahead of where the gene is located in a region before every gene. Every single gene has one of these. It's called a promoter. And that promoter region is typically made of a series of thymine and adenines. It's a series of thymines and adenines. So we call it the Tata box because it's thymine, adenine, thymine, adenine, thymine, adenine over and over and over again. Every gene has one. Yep. It occurs before the gene, so it's going to indicate to the enzymes where to start. It basically says, like, hey, shortly thereafter, a gene happens. So if you want to copy the whole gene, start here. Then you'll be sure you've gotten all of it. In this image, there is a big blue like circle around that promoter region. That is supposed to be an enzyme. You'll be shocked when I tell you that its name is RNA polymerase. Does that sound vaguely familiar? Where have you heard of a polymerase before? You've heard of DNA polymerase. Do you remember its job? It attaches complementary bases during DNA synthesis, right? Its job was to make the polymer, which is why its name is polymerase. RNA polymerase has the same job. Its job is going to be to open the DNA strand and make an RNA molecule that's complementary. Conveniently enough, this whole process starts at the beginning. In initiation. Yep. So it's all going to start in initiation where enzymes are going to bond to the promoter. The enzyme in question is going to be RNA polymerase. Welcome, friends. You didn't bring DF back? Oh, okay. It's like you just left him.
They just left me. It's like at the mall. I gotta find my own way home.
Well, as long as you have that emergency number on that like wristband, you're probably okay.
You know,
Call for help.
I thought about still writing that on my shirt in case I'm lost. And someone, like, if lost, if found, please return to Lexi.
You know what's funny? I actually have one of those for when I run.
In case of an emergency?
Yep.
How many of you have that on your phone? The ice number on your phone? If you haven't already done that, that's what first responders are looking for as they're going through your contacts. In case of emergency, IC, because otherwise they have no idea who these people are that are on your phone. Maybe they're close to you, maybe they're not. And instead of having to go through all of them, ICE tells them start with this person.
I just always hoped they'd call the one that said dad.
For me, my parents are in New York, and so it'd be a little harder. So you don't know who's closest that would be the contact person.
That we start at initiation, which is good because we always like to start at the beginning. It's a very convenient place to start. Where an enzyme is going to bond to the promoter region and it's going to unzip that little section of DNA. If we were comparing DNA to a cookbook, we said the cookbook is useless if we don't open it. Your DNA is useless if you don't open it. So it's going to be open. And I have bad news about the enzyme RNA polymerase. Do you remember the handicap that DNA polymerase had?
It can only go 3 to 5.
Could only read the template from 3 to 5. RNA polymerase suffers from the same inadequacy. So it can only move on the template strand from 3 to 5. But for those of you who are like, but I really hated that leading lagging thing. I don't want to deal with that leading lagging thing. Guess what? The lagging strand doesn't exist here. How many strands do you see being copied? One. So RNA polymerase is like, oh, it's really very easy. I just won't copy the 5 to 3 strand. I'll only copy 1/2. Because RNA is single-stranded, right? Done. So it reads the template from 3 to 5. And as soon as the template unzips and gets copied, it zips right back up. As soon as you're done using the cookbook, you close it. So DNA will close right back up. Much like our problem in DNA synthesis where we couldn't just attach to nothing, we will still attach to a primer. There'll be a short primer region that occurs before, and then RNA polymerase attaches the first complementary base. Once that happens, and that we call it an initiation complex, once we form the initiation complex, we move on to step 2. Elongation is super simple. RNA polymerase attaches complementary bases. If the DNA reads what will RNA polymerase attach? To C, it attaches A. To A, it attaches. To T, it attaches an A. C goes with. C goes with. G goes with. T goes with. And A goes with. Thank you. Yep, you. That's always tricky, right? You become such a habit for RNA or for DNA synthesis that you automatically want to do the thymine. You got really good at it, and you're like, thymine. And now we have to stop, though, because we're in RNA world, so it's uracil. That's all that elongation is. Then we're good. If you were to end something, where would you end it?
When you run out of stuff.
When you run out of stuff? How would you know that you've run out of stuff? RNA polymerase stops when it reaches a section of the gene called the terminator. It reaches a stop sign. So it stops.
The Arnold Schwarzenegger of the gene.
It is indeed. And every gene has one. We call it the termination sequence, or the terminator. Yep. So why do you have it? So the idea is your DNA possesses the instructions to make every protein that a body needs to make or do anything. So proteins, essentially, the chemical reactions that protein cause are the basis for all living things functioning and operating on a chemical level. The instructions to make those proteins are all in DNA. But DNA can't leave the nucleus. So, and proteins don't get made in the nucleus, proteins get made in the cytoplasm. So RNA gives us the way to get the information out of the nucleus and into the cytoplasm.
So it copies the DNA strand
Mhm.
using the template and it makes.
Exactly. So if we look at this, our product is actually not RNA yet. The product is called pre-mRNA. Hey, what's the mRNA? What was that? What's that one stand for? Messenger. And it's a good name because it really just sends the gene as a message from the nucleus out into the cytoplasm. But we're at pre-mRNA. We have to do a little editing, a little splicing, a little fixing here, a little fluffing up there, and it's very easy. The RNA gets edited in the nucleus. And let's just say it looks like, I'm going to color code in two different colors, regions that got copied. So here's a section of nucleotides that were copied. Here's a section. And here's another section. There's a little section of nucleotides. So all of these are just nucleotides that were copied, and we can label them. A, T, C, G, C, T. Oh, we're in RNA. Let's make that a U. Whew, that was close. So we have a sequence that have been copied, and they're color-coded. You'll see why in a second. First thing we do is put a hat on it. We call that hat a cap, and we put it on it to protect it. Much like you would put a hat on in the winter to protect your head, we're going to add a cap. And that cap is actually made of inverted guanines. They're guanines that are attached backwards. And that way they don't ever get incorporated into the protein. They're not read as instructions because the enzymes it encounters later knows that that's not part of the gene itself. And then we add a tail. And the tail has kind of a funny name. We call it a poly-A tail.